Hello, we're glad you've joined us for this live webinar, DNA and RNA Sequencing Sample Prep, Automating Simple to Complex Methods. I'm Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Beckman Coulter Life Sciences. This webinar is actually the second presentation in a three-part series on laboratory automation solutions for smaller NGS labs, those that are less familiar with automation who are interested in standardizing workflows and saving costs through improved efficiency and results. This year, Beckman Coulter Life Sciences is celebrating 30 years in automation as they continue to provide liquid handling solutions to help accelerate reliable results that drive scientific discovery. Beckman offers a full range of flexible and scalable biomech automated workstations and integrated systems to automate genomic, cellular, protein, and other workflows in a wide variety of research areas, including agriculture, cancer and infectious diseases, cardiovascular, forensics, and neurodegenerative disorders. For genomic applications, biomech automated workstations are complemented by a growing portfolio of nucleic acid extraction and purification kits built on the company's patented solid-phase reversible immobilization, SPRI, magnetic bead technology. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can pose questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. Before you meet the speakers, I want to advise that a series of poll questions will appear on your screen during today's presentation. You may select your answers, then close the poll questions by clicking on the X in the right corner. I now present today's speakers, David Horvath, MS, and Zach Smith, MS, who are both Senior Application Scientists for Beckman Coulter Life Sciences. David automates proprietary and third-party next-generation sequencing chemistries. He has a Master's of Science in Medical and Molecular Genetics from the Indiana University School of Medicine. David's career experiences span a broad range of life science fields, including forensic DNA analysis, medical diagnostics, and agricultural biotech. Zach automates proprietary and third-party next-generation sequencing kits. He has a Master's in Environmental Science from Indiana University. Previously, Zach worked at the university's Center for Genomics and Bioinformatics as a project scientist. David will now begin his presentation.
Hi, welcome everybody. Just to recap from the first series, we started off talking about the introduction to automating NGS workflows. Today's topic will expand on specific DNA and RNA sequencing applications, and then if you care to join us for the third segment, we'll be talking about automating NGS sample prep for niche applications. So a brief overview of the first webinar series reviewed the typical NGS workflow, and while all sequencing chemistries are different, the typical NGS workflow involves general processes such as reagent addition for various enzymatic reactions, temperature-controlled incubations, thermocycling, and reagent storage, bead-based cleanups with Ampure XP or Spry bead technology, and bead-based target capture. So what we do is we string those different modules together to create various sequencing workflows. The workflows vary in length and complexity, but in general they comprise those general processes. The next part of Part 1 webinar was to describe our application development process, from market research to building collaborative partnerships, scoping requirements and features of applications, and then bridging that into method development and optimization, followed by data generation and generation of application notes, posters, etc., followed by an internal quality review process, and then finally actually doing the application installations at the customer site with post-installation support. And we also had some software features and highlights specifically related to our new addition, Biomic Method Launcher and Guided Library Setup, as well as some data reporting functionality with our DART data acquisition and reporting tool. So I encourage you to check the webinar online if you missed part one. So today's talk will feature several DNA-seq and RNA-seq workflows that we have automated. And DNA sequencing primarily is related to whole genome sequencing or target capture. So whole genome sequencing is essentially the characterization of genetic variation or mutations across an organism's entire genome. It can include de novo sequencing or generating the initial reference genomes of new species. More typically, people would be using whole genome sequencing to do resequencing, which is mapping sequencing results against the reference genome in order to identify SNP variants, copy number variants, insertions, deletions, or large structural variants. Various applications for whole genome sequencing would include personalized medicine, predictive medicine, infectious disease strain identification, metagenomics, as well as others. So this is a typical whole genome sequencing workflow. You start off with fragmentation of your sample, followed by end repair, and then A-tailing, adding the adapter, ligating the adapter, which would include the sequence-specific adapter as well as a sample-specific barcode index for multiplexing sequencing reactions, and then typically followed by a PCR enrichment step. Alternatively, there is a tagmentation protocol in which the fragmentation, the ligation are all wrapped up into one step, followed by PCR enrichment. That's transposome mediated, and the nicking of the genome happens along with the ligation in one reaction. So various challenge points associated with whole genome sequencing are the ability to accommodate a wide range of sample inputs. So we have various automation solutions that provide, the user interface provides flexibility of sample input and adjust the protocol steps automatically. We also have a challenge with accurately size selecting fragments across a wide range of fragment sizes. So our protocol parameters will again automatically adjust and match appropriately to the sample input and fragment size selection. Using our proven SPRY magnetic bead technology facilitates high throughput processing in microtiter plates, providing consistent results from sample to sample and run to run. In addition, complex lengthy protocols require extensive hands-on time and sample manipulation. Beckman Coulter automated sequencing applications require minimal upfront setup with modular workflows that provide a balance of scheduling flexibility and significant walk-away time. Okay, next I'll describe two different sequencing applications to demonstrate how we've automated a whole genome sequencing process. Since this is a small labs campaign, we want to focus on the ability to maximize efficiency on our small platform, the Biomic 4000. And here we have an example of a Biomic 4000 application for the Illumina TruSeq DNA PCR-free as well as the nano-DNA methods. What we've done is combine both sequencing chemistries into one streamlined workflow. If you're familiar with both these chemistry protocols, PCR-free and nano are essentially the same protocol with the exception of the PCR enrichment step at the end, which is included in the nano-DNA protocol. So we've combined both those applications so that either workflow is available. What that does is allow the method to accommodate a wide range of input DNA. So 100 nanograms input for the nano protocol up to 2 micrograms of input for PCR-free protocol. So on the graphic to the right, it's our workflow. So the first column would be the fragmentation cleanup through size selection steps with a breakpoint corresponding to Illumina's recommended safe stop point. followed by method section 2, A-tailing, ligations, and ligation cleanups. For the PCR-free method, the process would stop there, and if you were running the nano method, you would have the option to continue on to the third section, which is the DNA enrichment, followed by the final PCR cleanup. So all of our methods include a user-friendly user interface that provides all of the modular options that are available for the manual protocol, as well as extra features that are associated with our automation protocol. The Biomac 4000 provides similar method option flexibility as you would expect in the larger NX and FX platforms. It's the optimal instrument platform for low to medium throughput laboratories, but it's also scalable to 96 sample workflows as labs make the transition to the larger NX or FX platform. It can accommodate up to 24 samples per run, anywhere from 1 to 24, and includes modular processing of protocol sections. So as I mentioned before, fragmentation through size selection can be performed as an independent run from the A-tailing and ligation reactions or subsequent PCR cleanups. There's flexible adaptive labware and transfer options. There's optional integrated thermocycling, which provides true walk-away capability, and also cost-effective disposable tip allocation. Method timings are consistent with or faster than manual processing as you scale up the sample number. DNA PCR-free workflow can be accomplished in approximately 5.5 hours, including 15 minutes of manual setup time. The nano portion of the protocol can be completed in approximately 7 hours, including manual setup of approximately 15 minutes. This is an example of the Biomac 4000 deck. It's highly flexible. For this particular method, we only use 3 tip boxes for the entire PCR-free or nano method for up to 24 samples. We have a mixture of single and 8-channel pipetting tools, which provide balance of precious reagent delivery and efficient cleanups. We have temperature-controlled reagent storage, ensuring robust reaction chemistry. And then the deck is flexible, allowing for optional integration of the thermocycler. We use stacked labware to maximize deck space when possible, and that allows the greatest number of samples to be processed with the smaller deck footprint. So here is final data from our automated Illumina TruSeq DNA PCR-free and nano workflows. So you can see consistency on the bioanalyzer data between replicates and sufficient library yield for PCR-free libraries of approximately 6 to 7 nanomolar. The same consistency for our nano DNA libraries and very high yields for those replicates, approximately 80 nanomolar. So highly sufficient yields for sequencing results. At the time of this presentation, we do not have Biomac 4000 specific sequencing data available, but feel free to reach out to an application scientist, either Zach or myself, or any of our field application scientists in the near future, and we'll be able to share that data with you. The second example is the Swift Biosciences Excel NGS 2S Plus method. Similar to the Illumina method, this is a PCR-free or PCR enrichment workflow. It provides either option. It has on-deck thermocycling, and 96 PCR-free libraries can be completed in approximately 4.5 hours. So this method is developed on the Biomac FX, and the associated data was generated using that platform, although the method is scalable to a Biomac 4000 platform, just as the PCR-free and nano methods were. The workflow here is broken into 2 different parts. Method section 1 includes the fragmentation, repair reactions, ligation reactions, and cleanup. Following those reactions, there's a safe stop point, and the second part of the method includes the PCR enrichment and cleanup steps. So you have the option to run the full protocol or just one or just the other. Again, this method includes a very customizable user interface. If you're familiar with the Swift protocol, There's multiple options for sample input range and fragment size. The combination of those two options drives different spry cleanup conditions. So the benefit of running through our user interface and our reagent calculator is that the spry conditions are automatically updated for the user. There's modular processing of the different protocol sections, as I mentioned, for the PCR-free library preparation portion or the PCR enrichment and cleanup portions. Data validation in our user interfaces ensure that only proper drop-down selections and inputs can be made. So if you see the start button at the bottom, if there's any improper entry, such as sample number greater than 96, then there will be messages down where the start button is indicating what needs to be corrected before the user can execute the run. There's options for on or off thermocycling, as well as the training mode that reduces time to optimize and test the method or train new users. In addition, we also have flexible adapter labware and transfer options. We went over in a little bit more detail in the first webinar about our Biomech Method Launcher feature. What it essentially is a simplified user interface for executing and launching a method. The Swift method, it was one of our early methods that was developed with this new software capability. And some features that Biomech Method Launcher provides are a guided labware and reagent setup, which saves time and reduces setup errors. It steps the user labware by labware through the deck setup and provides instruction for reagent configuration in troughs or reagent racks specific to the options selected in the user interface. During runtime, the information tiles provide real-time run updates, and it also has the capability to be run remotely or monitored from any web-enabled device. So this slide compares manual and automated library yields. So you can see from a wide range of inputs, 100 nanograms on the left down to 100 picograms on the right for E. coli genomic DNA input. There's consistency between the manual and automated library yields. This summarizes our sequencing data of the genomic DNA control. As you can see, we tested variable fragment lengths. There's 200 and 350 base pair inserts with a wide range of sample inputs ranging from 100 picograms to 500 nanograms. And you can see that the percent alignment is extremely high with very low PCR duplicates. We also tested FFPE material with various fragment insert sizes. Again, having sufficient yield, 4 nanomolar yield up to 27 nanomolar yield, depending on the condition of the FFPE material, and resulting in high alignment and very low PCR duplicates. So the next example would be related to targeted sequencing. So types of targeted sequencing are amplicon sequencing, using PCR to amplify specific target regions, or hybridization capture, where you will hybridize target-specific probes followed by typically streptavidin bead capture. Various applications for targeted sequencing would be cancer screening panels, exome sequencing, or forensic STR analysis. Targeted sequencing challenge points. For various targeted sequencing applications, there's a small number of samples which can expand rapidly with multiple amplicon reactions or restriction enzyme digestions. The automation method will simplify the replication of sample reaction setup and also manages and tracks data associated with each well. Reagent volume calculations can be tedious and can fail if improperly calculated. And through the use of our dynamic reagent calculators, those calculations are performed automatically based on the user selections made during run option selection. Also in capture protocols, there's typically long overnight hybridization reactions, so that creates a lot of downtime on the instrumentation that you don't want to be held up during those long hybridization reactions. So our sequencing applications provide modular workflows corresponding with the manufacturer recommended stop points or long incubations, which increase instrument productivity and multi-application use. So our systems can be are broken up into modules so that you can perform other tasks on the instrumentation while samples are being incubated for long periods off deck. So our application example for target capture is the Agilent Technologies HaloPlex protocol. This was automated on the Biomech 4000, and it was automated for the ion torrent platform chemistry. So the Agilent HaloPlex protocol is a relatively simple protocol when you look at these slides, but it actually is a little bit more complex from a sample handling standpoint. So essentially, the first step would be to digest and denature your sample DNA with a series of restriction enzymes, followed by probe hybridization to the DNA targets of interest, followed by purification and ligation of the target regions, and then a final amplification enrichment by PCR. So this slide kind of shows the complexity of the restriction enzyme reaction setup. So for every sample, there's 8 restriction enzymes that have to be processed. So you can imagine the amount of pipetting that occurs for setting up multiple reactions for each sample. So the automation alleviates this pipetting intensive setup for restriction digestion and also removes the potential for human error and is able to provide more consistent library preparation. Again, there's options for use via our user interface. The modular protocol provides more effective time management and flexibility around overnight incubation steps. And it's an error there. It actually can process anywhere from 1 to 12 samples with 8 restriction enzyme reactions for a total of 96 reactions per plate. The automated reagent calculator provides master mix and reagent volumes based on sample number and other user options. So this slide demonstrates the consistency of the Biomech 4000 and manually generated restriction fragments. As you can see, with the 2 restriction digests there, that the Biomech 4000 and manual look consistent with effective restriction digestion. This slide shows consistency of the biological sample prepared manually and on the Biomech 4000, and you're generating the same electrophoresis profile. This is done using the fragment analyzer from Advanced Analytical. Okay, comparisons of library size and yield are consistent from the manual and automated prep. And sequencing results provide 80 to 100 million reads with an average read length of 100 base pair and a sequencing depth of greater than 400 to 500 per sample. With approximately 101 million reads, we have approximately 80% on-target reads, and that's extremely consistent between the manual and automated prep. So our uniformity and our on-target percentage between the manual and Biomech 4000 can be considered comparable. Okay, and I want to just summarize the Agilent Technologies HaloPlex protocol in the Biomech 4000. It increases the accuracy of library preps, reduces human error with samples, requires fewer library failures than with manual preps, even with an inexperienced technician. The lower yield that we saw with the automated prep does not affect the sequencing ability or results. It was successful with FFPE DNA, provides cost-effective, time-saving solution, and increases productivity. And this development of the Agilent protocol, HaloPlex protocol, was done in partnership with Dartmouth Genomics and Molecular Biology Corps. So thank you to their team. And I would like to then hand off the rest of the presentation to my colleague, Zach Smith, who will tell you about some RNA sequencing applications.
Excellent. So thank you very much, David. Thank you all for hanging in here so far with us. I just wanted to start by talking about how we discussed, getting at the genome. How about the transcriptome? So The last half of this talk is going to be about RNA-seq applications that we've automated. The transcriptome is an interesting and wild place. RNA comes in a variety of types, include coding RNAs, your traditional messenger RNAs, and then you've got, it seems like an ever-expanding family of non-coding RNAs, including ribosomal RNAs, transfer RNAs, long non-coding RNAs such as this and hot air, and then a variety of small non-coding RNAs, including microRNAs, PV-interacting RNAs, and small interfering RNAs, just to name a few. And it seems like this list keeps getting longer the longer the deeper we go into the genome, or into the transcriptome, excuse me. So because you have all these various species of RNA, the tactics that you would use to get at them from a sequencing standpoint can vary a little bit on protocol. And so what we're going to go over today is specifically an mRNA sequencing workflow, a total RNA sequencing workflow, and a small RNA sequencing workflow. So mRNA sequencing specifically targets mRNA through the use of a poly-A enrichment technique using oligo-DT labeled beads. Basically what you're doing is you're hybridizing your oligo-DT bioassimilated beads to the poly-A tail, and using those to fish out the messenger RNA from your total RNA pool. Obviously, you can't use it with prokaryotes because they don't have poly-A tails by and large, but it does work well for mammalian species, and it works quite well across a wide range of species. So I just wanted to direct your attention to I guess it would be my right. This is the NEBNext ultra-directional RNA workflow for Illumina. So as you can see, the first thing we're going to do is we're going to do our poly-A capture using the oligo-DT beads. Then we will fragment the resulting messenger RNA and prime it for reverse transcription, and then make our double-stranded cDNA after that. Following which, the protocol is very similar to the DNA Seq workflow that David described earlier. We're going to repair ends, and we're going to do A-tailing. In the case of the NEB chemistry, we're going to be doing a user excision because their adapters have an interesting hairpin shape to them. And then we'll amplify the libraries. And that's pretty much the entirety of the mRNA sequencing workflow. So just as an example method, we're going to, and some of the challenges that we would typically see in mRNA sequencing, is a combination of RNA from a variety of sample types, including prokaryotic samples. How are you going about dealing with that? Ensuring good removal of uninteresting/contaminating RNA species, such as ribosomal RNA, which can constitute the vast majority of any total RNA sample, and ensuring uniformity in the sample processing. So how we've gone about doing that is we have a method for the NEBNext RNA ultra-directional kit that allows the use of either total RNA in combination with your poly-A enrichment, or using pre-purified RNA samples that you've used some other kit off-deck to use, such as a Ribo0 kit. So that allows you to get an RNA from your prokaryotic samples. For removing uninteresting/contaminating RNA species, we're using a combination of orbital shaking and tip mixing for efficient mRNA capture, which results in good RNA removal using poly-A capture. And then we have the ability of integrating an optional T-robot thermocycler, as David mentioned earlier, onto our decks. And that really gives us the most uniform incubation temperatures across the plate, which results in more uniform libraries. It also gives you maximized walk-away time by allowing the thermocycling to occur on deck. So this is the automation solution we've developed for the NEBNext RNA ultra-directional kit from New England Biolabs. This workflow and user interface and the data that I'm about to present has been largely developed on the FX, although we do have a customer site that's running this on a V4K. So very briefly, we're just going to go over and touch on the user interface. It's very similar to the ones that David presented. It gives you a variety of options, including the ability to specify your number of samples, whether or not you're going to use an on-deck thermocycler, what parts of the method you're going to process that day, and then a variety of options around how you're deploying your indices and what volumes you want to elute the final library in. The method itself is broken up into basically two large sub-methods. So over here we'll have the method that runs from total RNA all the way through to double-stranded cDNA cleanup, and that would include our mRNA purification, fragmentation, and priming, first strand and second strand synthesis, as well as the cleanup. And then the second module would include basically the library prep part of the workflow. So taking the double-stranded cDNA and subjecting it to end repair, A-tailing, adapter ligation, cleanup, and PCR enrichment. So all told, this method can do up to 96 samples in about 9.5 hours. We have some manual data from NEB that suggests that trying to do that many samples manually could take as long as 21 hours. So we've recognized significant time savings on this method using the FX. Obviously, the method runtime would be slightly longer on the B4K, but if you're processing fewer than 96 samples, it should be fairly comparable, I would think. Okay, so let's dive into some data. So for this experiment, we did a series of library preps using Agilent Universal Human Reference RNA at both 25 nanogram and 100 nanogram inputs. We had a series of, I believe, 3 to 4 technical replicates for each sample. And I'm just presenting some bioanalyzer traces of the finished libraries here for both the 25 nanogram replicate 1 and 100 nanogram replicate 1. And as you can see, we don't really have much in the way that we don't actually have any adapter dimers that we can visualize here, and then the yields look quite good. So all 6 technical replicates were sequenced on a single HiSeq run, and the data was analyzed in doing the biolabs. This graph here is showing actually the technical replicate reproducibility between our 2 input amounts. So on the graph on my left, you'll see a transcript expression correlation graph for the 25 nanogram technical replicates, where we're looking at the fragments per kilobase of exome per million reads mapped, the FPKM ratio. And what we find here is that when you're comparing those numbers across the transcriptome, we're seeing very good reproducibility between our 2 technical replicates for both the 25 nanogram input on the left and for the 100 nanogram input on the right. We also, the bioinformaticians at NEB also did a number of other analyses on the data. So one of the things we would like to know is where on the where in the genome are the reads mapping to. And as you can see here on the left, we've got the 25 nanogram reads are the vast majority of them are mapping to coding regions, which means we are doing an efficient messenger RNA pull down. And if you look at the graph on the obviously we do have some small amount of ribosomal sequence left, but it's less than 5%, and we do have a small amount of intronic regions, which means we're also capturing non-mature messenger RNAs as well. The graph is pretty much replicated for the 100 nanogram input on your right as well. On the bottom of this slide, we're looking at transcriptome coverage. So this is a determination of where the reads are mapping along the transcript from the 5' to the 3' end. Ideally, what you'd like to see is a uniform distribution of reads, which would indicate that our reverse transcription is not biasing the sample one way or another, and that the sample itself is of good quality. We'll talk some more about that in the next example method, because I have some data from that would be interesting. And the other thing we wanted to know is, for library complexity, how many PCR duplicates are we seeing in these libraries? And it's terribly not surprising that the 100 nanogram inputs, which are the green bars on this chart, had significantly fewer events of PCR duplication than the 25 nanogram inputs. Generally, with any NGS kit that you're working with, the more sample input you have for the library prep, the better the library will be. I realize that some people are limited on just the type of sample and what they're able to get, but more input will give you a better library, typically. The other thing we wanted to look at was the input correlation between the 25 and 100 nanogram inputs. So this is, again, another FPKM ratio chart. And we see very good correlation between the 25 nanogram inputs, which means that our input material is not biasing the results that we're getting, at least as far as this metric is concerned. Okay, so we talked a little bit about messenger RNA sequencing. The next thing I want to touch on is what is referred to as total RNA sequencing. So the idea here is that we're interested in as many different species as we can capture. So this would be our messenger RNAs, this would be our long non-coding RNAs. But the problem with this is that you've still got our ribosomal RNA, and in some particular sample types, you've also got other high abundance transcripts like hemoglobin that you would be wasting your time sequencing, and then you need to get rid of them somehow. So the whole idea behind this particular workflow is instead of using the oligo-DT beads to fish the messenger RNA out of the sample, what we're doing is we're using some sort of bead capture or some sort of hybridization event to hybridize the high abundance transcripts and remove them from the pool and then sequence everything else. So in the case of Illumina sequencing technologies using the Ribozero kit, it's actually a bead-based hide capture. New England Biolabs uses just probes that then bind to the high abundance transcripts, and then those are digested away. Those are two very good ways of getting rid of those high abundance transcripts that would reduce the effectiveness of your sequencing experiment. The other thing that's great about this particular way of doing things is that it works well with RNA species or RNA from prokaryotes because you're not relying on the poly A tail. But one thing to keep in mind with these sorts of capture experiments is that depending on, the capture probes are designed against a certain collection of model organisms, and if you're working with a model organism that is not one of the canonical 13 from the NIH, so you're not working with humans, mice, or rat, or Arabidopsis, for example, you might see a reduced efficiency in the rRNA or the high abundance transcript knockdown based on the fact that we're working on sequence similarity to do these hybridization events. So just something to keep in mind as you go about doing these sorts of experiments. So challenge points for this would be accommodating RNA, again, accommodating RNA from a wide variety of sample types, including non-model organisms, and ensuring good removal of your ribosomal, your high abundance transcripts. So again, for this particular solution, our method design allows for the use of any of the 4 Illumina Ribozero depletion kits that are currently available, and for the orbital shaking, so for the high abundance transcript removal, we're relying on a combination of orbital shaking and tip mixing to make sure we get good bead mixing and resuspension for good rRNA removal. So this is the example method we're going to use for this. So this is the Illumina TruSeq stranded total RNA method, and we've developed this one again on the FX, but we do have customers that are using this on smaller platforms. Again, the workflow is very similar to the messenger RNA, the mRNA sequencing example for the Illumina, the NEBNext stranded NEBNext ultra-directional kit. But the major difference is the ribosomal bead removal rather than the poly capture. So we conducted 2 sets of sequencing experiments with this in collaboration with Dana-Farber. So they were looking at, basically, we had two experiments, one with 4 samples using FF or fresh frozen material, and one is FFPE. And those were only 4 samples, but they were sequenced to a depth of 150 million reads approximately for each sample. And then we had a smaller experiment with 20 universal human reference RNA samples that were sequenced to a depth of approximately 10 to 25 million reads. And then this is a graph showing the alignment that you're seeing here for the reads after they were sequenced and mapped back to the human reference genome. As you can see, in both cases, we are in the fresh frozen versus the FFPE. We had slightly higher rates of unaligned reads for the FFPE samples, but not too bad on balance. In this slide, we're showing the abundant reads. So we're looking for contaminants in this particular slide here. So as you can see on the graph on the left, when we're looking at percent adapters, or we're looking at reads that are mapping back to mitochondria, or 5S ribosomal human DNA, or human ribosomal RNA or poly A, we are seeing extremely low rates of mapping to any of these contaminant reads in the fresh frozen or in the fresh frozen samples. We are seeing slightly higher mapping reads back to these contaminant reads back in the FFPE samples. But if you note on the graph, on the axis, on the y-axis of this graph, none of these readings get above 4.5%. So that means the vast majority of our reads are actually mapping to the targets that we're interested in getting at. And the same was true with the universal human reference samples. And actually, the data was somewhat better when we were mapping to less than 2% contaminants. This chart is kind of busy, but I'm going to summarize it by saying that we have fairly uniform median insert sizes for the universal human reference, as you see on the 20 or so UHR samples, and for the FFPE and fresh frozen as well. We do see higher rates of duplication in the fresh frozen samples as opposed to the FFPE samples. And then the third column is our strandedness. So this kit employs, as well as the NEBNext kit does this as well, employs a DUTP strategy for marking the strand that's complementary to the RNA molecule, which means we're maintaining the strand information of where those reads, or which strand of the DNA those reads were transcribed from. And so in this case, we had over 97% of our reads were mapping to the correct strand, which was really quite good. And we did a little differential expression analysis on the FFPE versus the fresh frozen. We were seeing a slight amount of differential expression in between 50 and less than 100 genes, though, in the fresh frozen versus the FFPE samples. One of the other things we were really interested in knowing is how, as far as alignment and coverage, how were those reads comparing versus for the fresh frozen versus the FFPE material. So again, we're going to look at a transcript coverage graph on the left side of the screen. So the blue arrow that you can see there is pointing to the FFPE samples. As you can see, they're slightly shifted towards the 3' end of the transcript, 3' end of the ideal transcript, and we're seeing slightly higher. We're seeing quite a bit higher intronic reads in the FFPE samples. So this just underscores the fact that certain types of RNAs don't work well with certain kits. So the stranded total kit does work fairly well for fresh frozen. It works well for fresh RNA, but it could be better for FFPE derived samples, and this is part of the reason why Illumina developed the RNA Access Kit, was to help get around some of the problems, some of the challenges with working with FFPE derived samples. When we're looking at our universal human reference, again, when we're looking at the transcript alignment to the ideal transcript, and we're looking at coverage controls, we're not seeing really any difference based on the number of inputs. So the graphs going from the left to right are from 50, 100, 200, and 500 nanograms of input material, and we're seeing fairly consistent data between those various input types or input amounts. Okay, so we've talked about mRNA sequencing and we've talked about total RNA sequencing. The next thing I wanted to discuss briefly was small RNA sequencing. So small RNAs are very tricky to get at. The kits that are developed around them, instead of doing some sort of pre-purification and then fragmenting the samples and priming them, they actually instead use a direct ligation strategy where we are ligating adapters directly to the small RNA molecules. And in part, this is to make them large enough that we can manipulate them with what I would prefer standard NGS techniques with like bead-based, sprite captures and whatn't. So this is the NEBNext small RNA workflow, and what we're doing is we're simply ligating on an adapter to the 3' end of the small RNA, and then the 5' end, and then we're going to land our our primer for reverse transcription, and then we are going to go ahead and do our first transsynthesis, and then do PCR to finish building the library. This is extremely typical of most small RNA workflows. So because we're doing direct ligation to RNA molecules, that is a challenge for even someone who's doing it manually. And the reason why is that the RNA ligases that are being used the efficiency of the ligation reaction drops dramatically if you manage to regain secondary structure in either your adapter or the target that you're trying to ligate to. So you need to heat denature the samples to make them linear, and then you need to keep them cold while you're working on them to make sure that you reduce the amount of secondary structure formation. So for our automation solution, we utilize on-deck cooling outs and labware that basically cool blocks to reduce the incidence of that secondary structure reformation, which ensures higher ligation efficiency. One of the things that's particular to the NEBNext small RNA kit is that they have developed a protocol for using our bead-based sprite technology for doing the size selection on the final library. However, they also recognize that you might want to use a gel cut or a pippin prep to do that final size selection. So in the automation solution, we've developed a method to where it does any one of those options. So this is our example method. So this is the NEBNext small RNA library prep kit for Illumina. The workflow has been developed, has been broken up into 3 major modules, the blue, red, and green. So we're doing our 3' adapter ligation, our RT hybridization, and 5'. And then we'll do our PCR amplification, and then our post-PCR purification and site selection. So this maintains all of the kit's stopping points. This solution was developed for the P4K and for the Bionec FX, and we have the data I'm going to be presenting today is from the Bionec 4000. So it can process up to 24 samples, and we give you a variety of options, including specifying how much you want to dilute the adapters and primers already. So if you're working with a sufficiently large amount of material, you might not want to do that. Or if you're dealing with a RNA sample that was derived from a microRNA enriched environment, such as brain tissue, then you might not want to dilute your adapters and primers. But if you're dealing with very dilute sample, or you're dealing with a sample that comes from a tissue that's not known for enriching small RNAs, then you might want to dilute your adapters and primers to reduce the incidence of adapter dimer formation. And so we give you that option right in the method. And then we can also give you the option of whether or not you want the instrument to deploy the index barcodes automatically, or if you want to specify a file, do that yourself, and what kind of post-PCR size selection option you want to perform, either NQXP based or pip and prep or some sort of off-deck gel cut. So this is a bioanalyzer trace overlay of a small RNA library before and after the bead-based size selection that's available in this kit. As you can see, the blue line here, this is generated from Agilent microRNA control RNA. So the blue lines, the blue traces here are in the background, the ones with multiple peaks are before size selection. And then after size selection, we see the red trace here, which has our approximately 120 or so base, 140 base pair product that we're interested in sequencing. So after adapter trim, so we sequenced, the samples were sent to NEB for sequencing and analysis. After we did a MySeq run, we then trimmed the reads back. MicroRNAs tend to range anywhere from 18 to 32 nucleotides, and so we're getting our small RNAs of approximately 22, which is right where we're hoping to see them. For RNA from colon, brain, and the Agilent microRNA control, and we did this at 2 input amounts, either 500 nanograms or 100 nanograms. So when we used base space to do the sequencing analysis here, and as you can see, we mapped the reads back to the reference genome. In this case, 20 to 30% of it was mapping back to 2 various microRNAs. We did see abundant reads in the human ribosomal primarily, but we did see a variety of, somewhere between 20% approximately. There were others, including peewees and lynx and the other small RNA species that you would expect to find. And this is a differential expression heat map based on the mRNA and isomer expression from the NEBNext small RNA libraries. So in this particular heat map, green indicates a high level of expression and red indicates low levels of expression. Use DESeq2 to perform the differential expression analysis, and completely unsurprisingly, we found that microRNA that was typically upregulated in brain tissue was upregulated versus the Agilent control, and likewise with the colon. But it was nice to see that results that we should have been getting when we were doing these comparisons we were getting. So with that, I just wanted to add a brief plug for our NGS splash page. So visit us at info.beckman.com/NGS for more information for our automated solutions. And thank you for your time. We'll be happy to take any questions now.
Thank you, David and Zach, for your presentations. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. David and Zach will answer as many questions as time permits. And the first question is, any automation for ion torrent RNA-seq library prep workflows yet? Whole transcriptome, small RNAs, AmpliSeq?
Please. Can you repeat the question, please, Judy?
Yes, of course. Any automation for ion torrent RNA-seq laboratory prep workflows yet? Whole transcriptome, small RNAs, AmpliSeq?
We're currently in the process of developing an AmpliSeq solution for the FX. We did show some ion torrent data in this presentation for HaloPlex, but other than that, no. AmpliSeq has been the one we primarily have been working on lately.
The higher NW peaks in the small RNA-seq that are purified away, are those due to concatenators of small RNAs or adapters? Sorry, concatenators?
Sure. No, are you in the small RNA data, you're referring to the overlay graph. The ones that are purified away are larger RNA fragments in the control RNA from Agilent that were ligated successfully. For the RNA ligation, basically you need a 5' hydroxyl and a 3' phosphate on the RNA molecule that you're ligating to. And you will see that sort of end combination happening occasionally, so those will get amplified because they have been converted into libraries successfully. So that's part of the reason why we're doing the size selection, is to zero in specifically on the size fragments ranging from 130 to 140 bases in length.
What applications do you currently have for Biomet 4000?
So for the Biomet 4000, we have, obviously we showed the TruSeq Nano/PCR-free method for the Biomet 4000, NEBNext small RNA from New England Biolabs. We've got a variety of PCR setup and extraction chemistries as well on the Biomet 4000. And we're in the process of expanding that portfolio. We do have a number of customer-developed or customer-implemented methods on the Biomet 4000. I don't have the list in front of me right at the moment, though.
Okay, now we're just going to pause for a moment. Let's see if more questions are coming in. And while we're waiting, I would like to once again thank David Horvath and Zach Smith for their presentations. Do you have any final comments?
Not at this point, other than thank you for attending.
Yeah, thank you again for everyone for showing up. It's good that you were able to take some time out of your day to listen to our talk.
Okay, great. Well, I see we have another poll up. And so while people are answering the poll, I would just like to thank the audience for joining us today and for their interesting questions. We would also like to thank our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. As a reminder, this webinar is the second in a three-part series. Please save the date for the final upcoming presentation. Part 3, Mission Possible, Automating NGS Sample Prep for Challenging Samples and Niche Applications, will be presented on July 28th. Registration is now open for Part 3, so feel free to visit the LabRoots website to confirm your attendance today. This webinar has been approved for continuing educational credit. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through December 23rd, 2016. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.